Okay, so I'm Harrison. The title of my talk is The Network Architecture Metabolism on Earth. Tessa did a good job doing some introduction to networks, and I'm going to tell you why they're relevant to metabolism. So basically what I mean by this is we're studying different types of metabolism, so metabolism of individuals, metabolism of communities, and you can represent metabolism as a chemical reaction network. And when I say network architecture, architecture just refers to the structure of the network, so just the different properties of the network itself. This is work I do with the postdoc in our group, Hanju, Jason, Raymond, and Sarah Walker, who are professors in the group, and then eLife, which is our research group, which will have a few talks this afternoon, helps a lot with input and feedback and stuff. Okay, so this is really big. For this talk, this is basically what I want to talk about today. So all life shares a common core metabolism. What I'm interested in is that chance or necessity? Is that required if you played the tape of life again? Would you always get only one dominant metabolism on Earth, or would you get a couple origins of life that can coexist with each other? What we also want to do is quantify the structure of Earth metabolism. So again, the metabolism of individuals, the metabolism of communities, all different types of life. What makes a co-evolved microbial community special? So if you have a group of organisms that co-evolve together, does that community look different than a group of organisms that you put in fresh and you're just watching them start to evolve, cohabitating in the same space? And then the last thing I want to address is, could we be living alongside a shadow biosphere? And so by this I mean, could it be that there is a second origin of life on Earth that we just don't know about? We haven't been able to detect either because we don't have the right techniques, or because it's living somewhere different than where we are, so it's physically separated. And that's what the bottom of this slide is showing. So you can have ecologically separate biospheres, or you can have ecologically integrated biospheres, or you can have biochemically integrated. And what I interpret that to mean is it uses different sets of chemical reactions to sustain life, but is overlapping physically in the same space as life as we know it. So starting at the organism level, we analyzed 21,000 bacterial genomes and 730 archaeal genomes. We also have 26 metagenomes from Yellowstone. And the reason we have metagenomes from Yellowstone, as probably a lot of you know, is because it's a really diverse environment. So it's really great to look at the mix of species across different pH and temperature ranges. And then we also looked at the biosphere level. So 3 different levels of biological organization. And to get to make a network from the biosphere, we basically just use this KEGG database, which has all the enzymatically catalyzed chemical reactions. And we just treat those as if they're all part of one physical thing, which they are, which is the biosphere. So first, I want to give you a little background on networks. Again, Tessa did a good job. This is a similar example. So I have a highway network here, and I represent cities as nodes, and then just the highways between cities are the links or the edges. In this kind of network for a highway, most of your nodes have roughly the same number of links. So you don't have any cities that have thousands of highways going out of them, and you also don't have very many cities where there's only one highway going out of them. And so this is what this distribution looks like if you plot the number of nodes with a certain number of links and then the number of links on the y-axis. So most of your nodes have the same number of links. This is as opposed to like an airport network where if you represent nodes as airports and then the traveling between the airports is the links. You do get hubs like Chicago, Boston, or LA, but you also have hundreds or thousands of regional airports that only fly to a few places. And so those are all right here where you have a lot of nodes with only a few links, and then your big hubs are down at the tail of the distribution where you have a few hubs with a large number of links. So you can represent lots of things as networks. This is an example that's really easy to understand. But we represent communities and individuals as networks as well. And so what we do is other people go out and collect samples at Yellowstone and got these microbial community samples from hot spring ecosystems. And then you can turn the gene fragments that you sample into chemical reaction networks. And so for those of you that want all the details, this is a little bit more detail right here. So you match the genes with the enzymes that they code for. You match the enzymes with the reactions that you know that they catalyze. And then you put all those reactions together into one big network, and that's what we analyze. So you do this for metagenomes, and you can also do this for individual genomes. The difference is we collect the metagenomes out in the field, and the individual genomes we just pull from Patrick, which is a database online of all these genomes. OK, so here's kind of the first results. This is just some network stats of real metagenomes and just real genomes and the biosphere. So here's a little key right here. And on this top plot, I'm showing the shortest path of a network. The shortest path is, I'm giving you a little example here. So if this is your network, the blue numbers are the shortest path from F to that node. And then if you do this for every possible combination of nodes in your network and you average it, you get a single number. And you get one of those numbers for each of the 21,000 bacterial genomes that we analyze, each of the 700 archaeal genomes, all the metagenomes, the biosphere, and then we plot it right here. And if you notice, everything kind of lumps together. There's no clear distinguishing shortest path for either the individual genomes or the metagenomes. The biosphere is way over here, but that's just because of the size of the network. If you notice, it has a similar shortest path. And this is just the box plot representation of that data. We also looked at the mean degree. We looked at a bunch of things. I'm just showing you a snapshot of what we looked at. The mean degree is just the number of nodes that each node is connected to, and you look at that for every node and you average it. And so here's another little example. You do that for each of the networks. And this time it's interesting because here you notice the real metagenomes in yellow stand out from all the individual genomes. So this particular measure can identify or distinguish communities from individuals. And you can see that here on the box plot, and then this is the biosphere keg on the right side. So then we did another thing, and this ties back to the question at the beginning of how do co-evolved communities look compared to communities that are just starting to evolve together, so maybe some kind of big perturbation and everything's kind of re-equilibrating in a community. Do those communities look different than communities that have been co-evolving for millions of years? And so the way that we decide to measure that is with something I'm calling synthetic communities. So what we do is we sample individuals. These are real genomes. These are the genomes that we pull from the databases online. And then you just take random samples of them, and you put them together, and you say, OK, this is a community. Even though they didn't co-evolve in the real world, they were completely separate. You just call them and then we're calling them a synthetic community. OK, so then we analyze that. This is the plots from the last slide that you just saw. So do you think you'd be able to distinguish synthetic communities from real co-evolved communities with network measures? Any inkling? Absolutely. Absolutely? So that's what we thought too, but it's really weird because you don't. And so this is only for two particular measures, again, but we did this for lots of different network measures. And you see the same thing over and over, which is these are the sizes of the synthetic communities, so just random samples of 10, 20, 30, or 40 individuals. And then these are the real metagenomes. And they all fall pretty much within the same range for these particular measures, the mean degree and the shortest path. And so that's kind of weird. So there are measures where they get distinguished a little bit more or they're a little bit more fuzzy. But in general, there's no clear measure that you can use to distinguish synthetic communities from real communities. So the last thing we wanted to look at was, OK, what if you had individuals that came together to form a community, and all these individuals relied on different core metabolisms? And so that's kind of a hard thing to study because we don't know of any other origins of life. So what we did is we tried to make a little toy chemistries from the real individuals. So what we did is we took the real individual networks, and all we did was we shuffled the labels of the compounds of the metabolites. And so effectively what this does is it keeps the topology of the network the same. It keeps all the links the same. But what it does is it mixes up just these node labels. And what happens is when you combine networks, even though they look the same when they're individual with the shuffled labels, when you combine them, the network's completely different because now different parts of the network are overlapping. And so again, this is to simulate what would happen if you had different core metabolisms of individuals forming a community together. What would that network look like? OK, and this is where it gets kind of weird. So on the left-hand side of these box plots, you can't really read the labels, but again, I'm showing you shortest path and mean degree. On the left side, we see all of the real communities, all the real individual genomes, and the biosphere. And we see the synthetic networks. Again, they don't really stand out. But starting here where it says nets 10 and it goes down to nets 70, these are these artificial communities. And the artificial communities really diverge from the architecture of the real communities and the real individuals. And this is kind of surprising. And they diverge in a certain way. This isn't the clearest legend, so I'll just walk you through it here. So on the left, again, we have the real individuals, the real community, the biosphere, and then the synthetic communities. And then all these nets labels are these artificial communities. And so with these networks, they have a particular type of distribution, which is similar to a power law distribution or a log normal distribution. And what that tells you is how the network's constructed, and it also tells you something about the robustness of the network. And so these networks are known from network science to be robust, these power law, these log normal networks. But then when you start looking at the artificial communities, you see that they're log normal, which is okay, but then as you grow these networks bigger and bigger, they become exponential. And these exponential distributions are less robust. And by robust, in this context, would mean you get some kind of mutation, you get some kind of perturbation to the community, and does the community recover or does it not recover? So you wouldn't see these communities recovering. And so we interpret that to mean, loosely, that a shadow biosphere is unlikely to be ecologically integrated with our biosphere. So maybe you could have certain types of shadow biospheres, like these ecologically separate ones. So maybe like in the deep earth, below life as we know it, there's other types of life living. And maybe there's integrated other biospheres, but the integrated biospheres would have to be using some different kind of information storage system than us. So instead of DNA, they'd have to be using something different. But what we think we can rule out is that you'd see other types of life that's using different core reactions to sustain themselves, living right alongside life as we know it. And that's what this last point is, driven by these results from these artificial communities. The other results that I just wanted to recap are, you see universal topological features for different levels of the biosphere, of different levels of biological organization, of individuals, communities, and the biosphere as a whole. And co-evolved communities are mostly indistinguishable from non-co-evolved communities. And so that's what we saw with the synthetic versus the real networks. But then the artificial communities are weird, and that's where we make this inference about the shadow biosphere. And then just recapping the points that I brought up at the beginning, is this chance or necessity that all life shares a common core metabolism? This seems to hint that this is necessity. You wouldn't see two types of life living, coexisting together in the same physical space. We quantified the structure of Earth's metabolisms. What makes a co-evolved microbial community special? Not much. There are some measures that they're distinguished a little bit, but most of the measures, they look pretty similar. Could we be living alongside a shadow biosphere? Well, yes, but not this particular type where you're biochemically integrated. So with that, thank you, and I'll take any questions.
We have time for a few questions.
Thanks, that was really cool. With regard to the not being able to distinguish the co-evolved communities from the ones you just throw together, isn't that definitely a signal to noise problem? Like if you start removing core consistent things, that signal will come up?
I'm not sure what you mean exactly.
So if you take away all core metabolism that's shared across everything,
sure,
and then look at them, I mean, I feel like your artificial part kind of shows that's what's going on.
Yeah, and I guess that's, in hindsight, that kind of makes sense, right? Because if you take a bunch of things that already share this common core, then they're going to look similar to things that are co-evolved with the same core anyways, right? Is that kind of what you're saying?
Yeah.
Okay, maybe not. We can talk after if you want.
Any other questions?
I have one really naive question on how you deal with the graph. So there are two kinds of nodes. You have reactions, and you also have species. You also have like the genetic sequences, I guess. So do you distinguish those two kinds of nodes, or do you shrink the reactions when you count the distance?
So we construct two different types of networks for the analysis, depending on the measures we're looking at. So one type of the network has nodes for reactions and nodes for metabolites, and then the metabolites are connected to the reactions if they're shared as part of the same reaction. The other one, we just do metabolites, and you connect those if those are part of the same reaction. And so depending on what measure we're looking at, we do analysis on one of those two types of networks. So for like the distance, like the shortest path, what we do is we do it for the substrate-substrate networks, so just the metabolite. Sorry?
In that case?
No, we don't do any weighted edges here. These are all simple graphs.
Any other questions? Okay, moving onward. Next up is Ben Intel.